Good morning, everyone, and I'd like to thank the organizers for the opportunity to speak with you today. So my lab works on breast cancer metastasis. Metastasis is the leading cause of breast cancer-related deaths, with more than 40,000 each year in the US alone. Metastasis occurs when cells from a primary cancer, such as breast cancer, will disseminate to distant sites, such as the lung, bone marrow, and brain, where these cells may then colonize, and this is a rate-limiting step in the metastatic cascade. And those cells that do colonize then can cause organ dysfunction, which can lead to death. Therefore, there's a dire need for new biomarkers and treatments of metastatic disease. So my lab studies the integrin alpha V beta 3 as a marker of activated stem cells. So alpha V beta 3 is a cell surface receptor that binds to extracellular matrix proteins. It is composed of two subunits, an alpha and a beta subunit. So we previously showed that alpha V beta 3 was a marker of activated mammary stem cells important for mammary gland development during pregnancy. But it's also important in cancer stem cells for properties such as tumor initiation. So here we see a 6-fold decrease in tumor-initiating cells due to beta-3 knockdown in a breast cancer cell line. And it's important for metastasis. So here we have these metastatic green nodules in the lung that decrease in response to beta-3 knockdown. So in addition to these properties, alpha V beta 3 positive cells also express cancer stem cell markers and are enriched for anchorage-independent tumor sphere formation, among others. So they really check the box with regard to cancer stem cell properties. Additionally, we show that they share gene expression similarities with hormonally activated stem cells in the mammary gland. Thus, we refer to them as activated cancer stem cells, or ACSCs for short. So ACSCs predict metastasis when in patient disease. So we examined 60 different primary tumors from breast cancer patients, which we stained for alpha V beta 3, shown here in blue, and the transcription factor slug, shown in brown. So here's a nice example of one of these double positive cells, where you have the beta 3 in the cytoplasm and plasma membrane, and the slug in the nucleus. So the first thing you'll notice is that these cells are a rare subset of tumor cells. Most of the cells nearby express slug but lack beta 3 expression. So we wanted to know if these cells predicted metastasis when we found them in patient primary tumors. And indeed they do. 84% of the tumors that had these cells went on to develop metastatic disease. And importantly, we found these in ER-positive and triple-negative breast cancers. So they're independent of breast cancer subtype. So this made us wonder if there was a heterogeneous cell line model available that we could use that had these ACSCs so that we could better reflect the situation in patient disease. So we settled on the HCC38 cell line as a good model system for these studies. HCC38 cells have been previously shown to be transcriptionally heterogeneous from the Pratt et al. Paper. They show that these cells have a subpopulation of EpCAM high and EpCAM low cell types. So we added alpha V beta 3 to this, showing that there's an alpha V beta 3 positive subset of each of the EpCAM high and EpCAM low cell types. Now I want to draw your attention to these EpCAM low alpha V beta 3 positive cells, which we've now characterized and show that they have properties of our ACSCs. So these EpCAM low beta 3 positive cells, if we inject them into mice, they are about 3 to 4-fold better than the other cell types in terms of initiating tumors. And we've also validated the presence of these cells in our HCC38 cell line using single-cell RNA-seq. And this was very gratifying to see using an unbiased approach that we could use or find similar cell types as we did with our flow cytometry. So these EpCAM low alpha V beta 3 positive cells also express cancer stem cell markers, are enriched for tumor sphere formation and self-renewal, tumor initiation, which I just showed you, as well as differentiation. So they really check the box with regard to all these cancer stem cell properties. So this cell line model better reflects human disease, allowing us to now analyze the genes and pathways that could be unique to our ACSCs. So one of the properties we've recently observed in our ACSCs is they are uniquely able to tolerate the stress caused by chromosomal instability, or CIN for short. So CIN is a hallmark of cancer. It is an important stress that limits transformation. It includes aneuploidy, as well as smaller copy number alterations, otherwise known as c-Jun. Notably, it does not include point mutations. The causes of CIN include persistent missegregation of chromosomes or incorrect repair of double-strand breaks. So this suggests that aggressive cancer cells must have a way of coping with this important stress. So we previously identified two genes that were specifically expressed within our ACSCs, and that's the gene TGF-bI, or TGF-beta induced, which is not to be confused with the ligand TGF-beta 1. This is a secreted extracellular matrix protein. And the transcription factor ZEB1, best known for its role in EMT. Now, we identified these through unbiased RNA-seq and functional screening. They were independently identified. However, we have now found that TGF-bI actually induces expression of ZEB1, so they seem to be related in a similar pathway. Independently, we identified the stress response gene set as a key feature of our ACSCs. And notably, ZEB1 has previously been found in normal mammary stem cells to enhance tumorigenicity by suppressing levels of CIN. So since we were unable to find an effect of our ZEB1 or TGF-bI knockout cells on proliferation or survival, we wondered if this pathway may be playing a role in CIN tolerance. So there are many different ways to measure CIN. We're starting here by looking at DNA damage using the marker phosphogamma H2AX to measure chromosomal instability. So here you can see nuclei that also have the DNA damage marker, and that these nuclei become more abundant in our TGF-bI knockout and ZEB1 knockout cells relative to control. And this is quantified here, showing about a twofold increase in phosphogamma H2AX positive cells in the ZEB1 knockout, whereas there's a more modest increase in the TGF-bI knockout in two different cell lines. We also examined micronuclei as a measure of CIN, and we found again an increase in micronuclei associated with both our TGF-bI and ZEB1 knockout cells in two different cell lines. And just to be thorough, we use one more measure of CIN. Here we're looking at copy number alterations, or c-Jun, using an Illumina SNP array. So these are volcano plots displaying SNPs that are either gained on the right side or lost on the left side, and the ones shown in red are statistically significant. So in both TGF-bI and ZEB1 knockout cells, we see enhanced levels of CIN. So both cell types display increased DNA damage, micronuclei, and c-Jun, all markers of CIN. So this suggests that the TGF-bI ZEB1 pathway we discovered may be important for maintaining CIN at tolerable levels. So we hypothesize that CIN tolerance may be a critical attribute of stemness. But here I need to pause and share with you some of the recent work that has described an alternative function for CIN in metastasis. Paradoxically, CIN has also been shown to promote breast cancer metastasis. In fact, it is required for metastasis in breast cancer cell lines. And this was found to be through inducing antiviral signaling through the cGAS-STING pathway. So in addition to causing stress and more aggressive cells needing to tolerate the stress caused by sin, increased sin can also have tumor-promoting effects through enhancing c-Gas sting signaling. So this led us to revise our hypothesis that ACSCs may now have higher levels of sin due to the fact that they can tolerate them, but also this could enhance the signaling downstream that might make them more aggressive. So to address this question, we examined our sorted HCC3 cell tumors, but this time we used a different measure of sin. Here we're using anaphase missegregations, which are actually the most rigorous measure of sin. So here's an example of a normal anaphase cell, what we found in our tumors. So these are the two different anaphase end plates that have nicely divided. And here's an example of two different types of missegregations. Here we have anaphase bridges that have formed between these end plates. And here we have some legging strands and possibly a bridge here that have also formed. So these missegregations can be quantified and expressed as a percent of the total anaphase cell. So this is what we're showing here in the tumors formed from all our different sorted cell types. And we see that the small tumors from the ACSCs have about twofold higher levels of sin relative to the other cell types. So this suggests these cells may be more sin tolerant. It's notable that the larger tumors from the same cells have less sin. So it suggests that this sin may in fact be transient and that the levels will decrease as tumors become more established. So we examine this possibility by looking at our freshly sorted cells where we measured sin using the sin gene signature. However, we noticed no difference in the basal levels of sin in these freshly sorted cells. So this indicates that this increased sin we're observing is probably specific to the tumor initiation process. And in fact, sin may be a critical barrier to the initiation of new tumors forming from these cells. And these ACSCs may be uniquely able to overcome this stress. We similarly analyzed sin using our sin signature score in many different breast cancer cell lines representing each of the intrinsic breast cancer cell types. Again, finding that there's no real difference in sin levels between these subtypes, with only a trend toward lower levels in the luminal B breast cancer cells. We further validated these sin levels using anaphase missegregations, this time staining the cells with DAPI to visualize nuclei. So here's an example of a normal anaphase cell, and here's a nice example of an anaphase bridge representing a missegregation. So quantifying these missegregations, we again see lower levels in both of the luminal B breast cancer cell lines and similar high levels in both the basal-like and the stem-like cell lines. So there are similar levels of sin in basal-like and stem-like cells. So then we wanted to know if stem-like cells might in fact be more sin tolerant based on our findings from the tumors. So for these studies, we used reversing, which is an MPS1 inhibitor to induce sin. In our cells, we see about a twofold increase in sin upon treatment with 250 nanomolar reversing. It also induces formation of numerous micronuclei in these cells, which you can see by these little blue dots surrounding the larger cell nucleus. So using reversing to enhance sin levels similar to what we see in our tumors, we performed dose-response cell viability assays using two different stem-like and two different basal-like cell lines. And surprisingly, we observed a 100-fold difference between these cell types, with the stem-like cells being much more resistant to sin and the basal-like cells being much more sensitive. So this shows that this response to sin is very heterogeneous, differing in different cell types. So now we want to know what the mechanism was that was regulating the sin tolerance in ACSCs. Tolerance. So for these studies, we performed phosphoarray profiling from our sorted HCC38 cells. Each of these phosphoproteins is shown here in duplicate on a dot plot. And only one of these phosphoproteins was observed to increase specifically in our ACSCs relative to the other three populations, and that's circled here in red. And this is phospho-c-Jun. And we further validated increased phospho-c-Jun, specifically in these EpCAM low beta-3 positive cells. We also observed increased phospho-c-Jun in the stem-like claudin low cell lines, and we show that it's required for tumor sphere formation using a JNK inhibitor, which blocks phospho-c-Jun, and showing that both of these JNK inhibitors block colony formation. So what are the c-Jun dependent genes that are important for SYN tolerance? So we identified AXL as a candidate gene that's enriched in ACSDs downstream of c-Jun, and we did so by looking at the intersecting genes from 3 different gene sets, including c-Jun dependent genes from breast cancer, our CSC genes, and SYN-induced inflammation genes. These all converge on AXL as the only gene common to all 3 gene sets. We validated AXL mRNA expression to be increased specifically in these EpCAM low and beta-3 positive cells. And we further show the protein is also enriched in these cells as well. And AXL is specific to the stem-like claudin low cell lines. We further showed that AXL is a c-Jun dependent gene. 2 different JNK inhibitors reduce AXL mRNA expression, and they also reduce protein expression in 2 different cell types. And finally, to examine whether AXL was important for SYN tolerance in our stem-like cells, we constructed an AXL knockout cell using CRISPR-Cas9. Compared to control, AXL knockout had little effect on colony formation. When we treated with a low dose of reversing, this had little effect on the control cells, whereas the AXL knockout cells were much more sensitive. And this is quantified here, showing in 2 different cell lines that low doses of reversing that have little consequence in the control cells produce larger effects within our AXL knockout cells. So they're more sensitive to SYN stress. So just to summarize, we made the unexpected discovery that basal levels of SYN alone cannot distinguish stem from non-stem cancer cells, and that the response to SYN is heterogeneous, with ACSCs better able to tolerate this stress compared to basal-like cells. We further showed that this was due to enhanced activation of c-Jun and AXL stress signaling, and that this is a unique feature of ACSCs, making them more SYN tolerant. And this SYN tolerance may then allow greater SYN-induced inflammation signaling in our ACSCs, which will be the focus of our future studies. And I'd just like to thank the folks who did this work. The postdocs in my lab, Shanawaz Baba, primarily did the c-Jun and AXL studies along with c-Sun, and I'd like to thank our collaborators at UCSD. Olivia Harris-Mendy and Rich Klimke's labs were both helpful with the bioinformatics studies, and our collaborators at the Huntsman Cancer Institute and OHSU. Thank you, and I'll take any questions.